Hi all, welcome back to our channel. This is Dechama taking you all to yet another exciting and informative video. Today's topic is a very famous gene editing tool named CRISPR. Before getting into CRISPR, let's see what gene editing tools actually are. Gene editing tool is basically a mechanism wherein genetic material or DNA is being modified so that you can target a specific disease. So each and every disease in our body that is present is because of genetic manipulation which happens in our body. Now, if you target those genes or target those sequences where the mutation has happened because of which a disease has occurred, you can cure those diseases. So the basic motto of genetic tools or gene editing tools is to cure these diseases, is to make sure that those manipulations are done because of which you can cure a specific disease. Now, CRISPR is one of those tools. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now, this is a natural defense mechanism which is present in bacteria. What do I mean by this? Whenever a bacteria is attacked by a virus, it prevents itself because of this CRISPR mechanism which is already present inside its cell. Now, scientists have used this mechanism and modified it in such a way that it can be used to target human diseases as well. How do they do this? To understand this, we need to understand how the mechanism inside a bacteria takes place. So, to briefly explain about it, whenever a bacteria is attacked by a virus's genomic DNA is being cut and attached to the bacterial genomic DNA. Now, there is a sequence which is formed. And this sequence is being converted to CRISPR RNA. And that RNA is recognized by the molecular scissors, for example, Cas9 in the bacteria, which then cuts this viral DNA and hence does not allow the production of more viral DNA inside the bacterial cells. So, it prevents the bacteria from getting destroyed. This is the basic concept of CRISPR, and this concept is being attached to human diseases. How? Instead of cutting up the viral genes or viral DNA, scientists have modified it in such a way that the Cas9 or the molecular scissors recognize the genetic sequences of human diseases. Basically, instead of a viral DNA getting integrated into the CRISPR RNA, it is human disease genes. So these are recognized by CRISPR, and then they go and chop that DNA where this particular sequence is present, hence preventing the disease from getting any further and also finding out any mechanism of drugs that can target these diseases as well. So not just in humans but also for plant production where yield has been focused on to increase the yield of a particular product, also CRISPR has been used. Now, CRISPR has been targeting a range of diseases, including HIV, sickle cell anemia, Huntington's disease, different types of cancers, and even combating high cholesterol levels. But CRISPR still have a lot of ethical concerns. One of the reasons can be the use of CRISPR technology in germline editing, where you can edit the eggs and sperms of humans, gene editing in sperms and eggs to produce a particular offspring with a particular trait or with a particular genetic modification. So the effect of this on the individual being born can have both positive and negative effects. So that is where one of the ethical concerns can happen. Second is CRISPR technology may be involved only for a particular trait for a particular population. But then because there are chances of inbreeding, there might be a problem with genetic diversity, because of which these species might not be able to keep reproducing, or they might get to a point where the reproductive capacity of this particularly modified organism can stop, or they can lose the ability to reproduce. These are the ethical concerns. So there are a lot of safety issues, be it for plants or for humans. To combat these issues, National Academies of Science, Engineering and Medicine has put up a comprehensive set of rules and regulations. So basically there's a guideline. So any institution or any organization that wants to work upon want CRISPR or any other gene editing tools, these rules and regulations need to be followed. So there's a balance between research and safety. So this is all about CRISPR. See you all next time.